So, hello, continuing where we left off in the last episode. In the last episode, we got a basic overview of what eukaryotic cells are, and now we are going to learn about the structures within eukaryotic cells. So, let us now look at the individual cell organelles to understand their structure and functions. So, what is cell organelle? What are cell organelles? Organelles just literally translates to little organs. Cell membrane. The first cell organelle that we are going to learn about is cell membrane. Cell membrane is also known as the plasma membrane. So both the terms are used interchangeably. They mean the same thing, but it is different from cell envelope. Cell envelope is essentially everything that covers the cell is included in cell envelope. So that would include cell wall, cell membrane, middle lamellae, or anything that covers the cell is enveloped in the is a part of the cell envelope. So cell envelope is a broader term, and a part of the cell envelope is the cell membrane or the plasma membrane. So both of them are the same. Cell membrane and plasma membrane are the same terms. So the detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. So to go in depth into to actually look at the structure of the cell, they would have needed to look at the structure of the cell membrane, they would have needed a very powerful microscope. So the detailed structure only scientists could only study after the invention of the electron microscope. Meanwhile, before the discovery of the electron microscope, chemical studies on the cell, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. So now let's understand what these chemical studies are. Okay, first we'll understand what these chemical studies are. So let's just say, I'm just giving a brief example of how these chemical studies are done. Let's just say that you have a compound A. Okay, you have isolated a compound A. You have that compound A, and you notice that when you add, let's say, HCl to this compound A, then you get a unique compound B. Okay, then what you do, you take the cell membrane, from a cell, and then add HCl. So cell membrane plus HCl, you add HCl to that, and then you notice that there are a lot of compounds forming compounds, but there's also formation of B. So from that you can deduce, you can imply that there is a chance that compound A is also present in the cell membrane. So the cell membrane may be composed of compound A. So that is like that, with trial and error, with different compounds, with trial and error, they find the composition of the cell membrane. So that is what are chemical studies. Okay, now especially in the human red blood cells. Now why only in the human red blood cells? Well, that is because adult human red blood cells don't have many other membrane bound organelles. So think about it. In a usual eukaryotic cell, there is a cell, normal eukaryotic cell, and within that cell there are many membrane bound organelles also. So these membrane bound organelles, these membranes are different from the plasma membrane, right? These membranes are different from the plasma membrane of the cell. So what would you do if you want to study the plasma membrane of the cell? What would you do if you want to study the structure or composition of plasma membrane? Would you choose a cell that has other membrane bound organelles within it, or would you choose a cell that does not have any membrane bound organelles within it? You would obviously choose a cell that does not have that many membrane bound organelles within it, right? So that you can get a more accurate description of the plasma membrane. Because now note that scientists can't actually go into the cell structure. They can't, because this is before the invention of electron microscope. So now they're just doing chemical studies. So the best method would have been to take a cell that does not have any other membrane bound organelles within it. So that is why they chose human red blood cells, because it does not have that many membrane bound organelles within them. Now one more point, why does human red blood cells not have that many membrane bound organelles within them? Well, that is because it needs to carry a lot of oxygen. So lesser organelles would mean more of oxygen these red blood cells can carry. So that is the reason. So they did, and so scientists conducted chemical studies on RBCs, and with that they were able to deduce a possible structure. This is not the exact structure, because exact structure we can't know until we are actually going into and studying and looking into the structure of the cell and see. But this is just a possible structure. Chemical studies on human red blood cells enabled the scientists to deduce a possible structure of the plasma membrane. Okay, So these studies, means the chemical studies, showed that the cell membrane is mainly composed of lipids and proteins. Now what are lipids? Lipids are essentially hydrocarbon chains, right? They're just made up of carbon and hydrogen, and proteins are essentially amino acids, amino acid chains. Amino acids are made up of nitrogen and hydrogen. Okay, so nitrogen, hydrogen, and sometimes carbon also. So we are going to go in depth into understanding the structure of lipids and proteins in the next chapter, which is biomolecules. This is a good chapter. So for now, just understand that lipids are made up of hydrocarbons, and proteins are made up of amino acids. So now the major lipids are phospholipids. Now phospholipids are different from, means they are a type of lipid. So they have two things in it, phospho and lipid. So these phospholipids have two things in them. One, they have a phosphate group. And the second, they have a lipid group also. Okay, so lipid group would be the lipid group, which is the hydrocarbon chain, and this would be the phosphate group, which contains phosphate. Okay, so it contains a compound that has phosphorus in it. That is why it is called a phosphate group. So phospholipids that are arranged in a bilayer. So the phospholipids are arranged. This is one phospholipid. The whole thing, this is one structure of a phospholipid. Okay, which is having a phosphate group, phosphate head, and polar and phosphate head and lipid tails. Okay, so it is arranged in a bilayer. So means two layers are arranged like this. Okay, let's read. Okay, two layers are arranged. So also the lipids are arranged within the membrane with the polar head outwards and the hydrophobic tail towards the inner part. So the polar heads are outwards. So which are the polar head? This is the phosphate group. So now let's understand first we'll have to understand what polar and non-polar compounds are. You might probably know what polar and non-polar compounds are. Polar compounds are those compounds that have some charge within the compound. They have some charge separation within the compound. So let's say, for example, there is a compound A and B. Okay, and B is more electronegative than A. Then what will it do? If B element is more electronegative than A, it will pull the electrons in this bond towards itself, thereby gaining a partial negative charge because electrons are negative. And the A, since it's losing a bit of partial charge, it will gain a partial positive charge. Now due to which there are poles forming on within the compound, there are poles forming, and such compounds are called polar compounds because there are formation of poles, there is a charge separation. Now the magnitude, means the magnitude of the charge that is formed due to this charge separation is the same, although the type of charge is different. One is positive, the other is negative, but the magnitude, the amount is the same, right? As much as negative charge this B is gaining, exactly that much positive charge A is gaining, because the amount of charge is the same, right? That is why. So such compounds are called polar compounds. Examples of polar compounds are essentially water. Water is a very good example of polar compounds. Or you can take HCl. HCl is also an example of a polar compound. So all you have to notice is that in a compound, whether to understand they are polar or non-polar, you just have to know which is more electronegative. That you will understand as you are learning chemistry more and more. But for now, I'll just explain. Oxygen is more electronegative than hydrogen. Hence, oxygen gains the delta negative charge, whereas the positive part is hydrogen. Delta partially positive charge is hydrogen. Then let's move on to non-polar compounds. Non-polar compounds are those compounds that, let's say you have compound A and C. So A and C, or you can say A and A. Since A and A compound, the elements are the same, they will have the same tendency, the same power to pull the electron. Hence there is no, hence the electrons will just remain at the center. Hence there is no charge separation. So see, even here the net charge is zero. Net charge is zero, right? Because as much negative is forming here, that much positive is forming here. So the net charge is zero. But there is a charge separation within the compound. Whereas here there is no charge separation and the net charge is zero. Okay. So such compounds where the charge separate, there is no charge separation, there is no formation of poles. Such compounds are called non-polar compounds. Non-polar compounds. Okay. And example of non-polar compounds can be carbon and hydrogen, hydrocarbon compounds, right? So anything that has carbon and hydrogen, if it only contains carbon and hydrogen, they are non-polar compounds. Why? Because the electronegativity difference between carbon and hydrogen is very less. we are going to go in depth into understanding what electronegativity is and all those things, and which has more electronegativity, which has less electronegativity, but that would be in chemistry. For now I am just explaining it that carbon and hydrogen don't have that much electronegativity difference. That is why they are non-polar compounds. So anything that contains only carbon and hydrogen, they are non-polar compounds. Now what did I just say lipids are made up of? Lipids are usually containing hydrocarbon chains, right? Hydrogen and carbon. So that means lipids are non-polar compounds. Whereas these phosphate groups, the phosphate part of the phospholipids, the phosphate group is essentially a polar compound. So the phosphate part of the phospholipid is polar compound, whereas the lipid part is a non-polar compound. Now what you have to understand here is that like dissolves in like. Okay, so if there is a polar compound, then that polar compound will only dissolve in polar compound, and if there is a non-polar compound, then non-polar compound will only dissolve in a non-polar compound. So to be honest, I mean, this is just a general rule of chemistry, that like dissolves like. But the main point here is that usually what happens is non-polar compounds tend to be with non-polar compounds. If A is a non-polar, if ABX is a non-polar compound, then it will tend to be with another ABX or another non-polar compound only, because they like each other. Okay, and now because we are going to go in depth into why they are like that in chemistry, but for now you to just understand, you just need to understand that non-polar compounds tend to be with non-polar compounds, and similarly polar compounds tend to be with polar compounds. So as I said, lipids are non-polar compounds, so they will tend to be with non-polar compounds, and they will want to avoid polar compounds. And what did I say polar compounds are? Water or HCl. Usually in a surrounding of the cell, there are non-polar compounds. Like let's just say in humans, if you're taking a cell in a human body, most of the human body is made up of water, and water is what? A polar compound. So where would lipids be? Which are the lipid part? This is the lipid part. Where would they want to be? They would want to be away from the water, right? They would want to be away from the water. Whereas the phosphate group, since it is a polar compound, it would want to be towards the water, towards the polar compound side. Hence the polar part of the phospholipid will be towards the outer side, and the non-polar lipid part of the phospholipid will be towards the inner side, thereby giving this plasma membrane a bilayer structure where the lipid part is towards the inner part of the membrane and the polar heads are towards the outer part of the membrane. I hope you are understanding. Okay, now let's understand this, then only you will because without this explanation you would not get the feel of the topics. Okay, so let's just read this again. Okay, so these studies showed, the chemical studies showed that chemical cell membrane is mainly composed of lipids and proteins. Okay, the major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the cell membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment. Why is it protected from the aqueous environment? Because aqueous environment is polar, and non-polar would want to be with only polar, and non-polar would want to be with only non-polar and would want to avoid any polar environments. And since aqueous environment is a polar environment, hence the non-polar tail would want to avoid the polar environment, or avoid the aqueous environment. Okay, so in addition to phospholipid, okay, one more word, new word you have learned here, which is saturated hydrocarbon. Now what are saturated hydrocarbons? It's very simple. Anything that has, so let's just say you're going to learn all these saturated, polar, non-polar, everything in chemistry, but just because I need to explain this para to you, I'm going to go in depth into what these things are. Okay, so saturated hydrocarbons. So anything that has hydrogen and carbon atoms are called hydrocarbons. Okay, anything that has hydrogen and carbon atoms, only hydrogen and carbon atoms, they are called hydrocarbons. Now let's say you have two carbon atoms and you have a double bond between them. Okay, so since carbon valency is 4, valency of carbon is 4, it will have only only 2 hydrogens. But what is the maximum number of hydrogens that can be attached in this sort of a system? The maximum would be 6, right? Because if there was no double bond, if there was only a single bond in this system, then there would be maximum 6 possible hydrogens that are attached to this compound. Similarly, let's say you have a carbon atom. Okay, so what is the maximum number of carbon atoms, maximum number of hydrogen atoms that can be attached to this carbon atom? There can be 4 hydrogen atoms attached to it. So saturated hydrocarbons are essentially those hydrocarbons in which there is maximum amount of hydrogens attached to it. So this can be explained in another way, that saturated hydrocarbons will have only single bonds in them, whereas between carbon and unsaturated hydrocarbons will have double or triple bond between carbon and carbon, because that will be less than maximum number of hydrogens present in the hydrocarbon. So they are saying that this ensures that the non-polar tail of the saturated hydrocarbon. What are the saturated hydrocarbons? Lipids. Lipids are made up of hydrocarbons. As I said, they are made up of hydrocarbons. So the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. Okay, I hope you understood. I hope you got the feel of the para, because this is a very good paragraph. So in addition to phospholipids, membrane also contains cholesterol. Okay. So what all does the membrane contain? Lipid, protein and cholesterol. Later, biochemical investigation is the same thing as chemical studies. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. So proteins, so we already knew. So lipids, proteins, cholesterol and carbohydrate. Plasma membrane contains lipids, proteins, cholesterol and carbohydrate. Now what are carbohydrates? Carbohydrates are essentially sugars. It's just another way of saying sugars, right? I mean, sugars are a type of carbohydrates. Okay, we are going to learn in depth into what these carbohydrates, what these cholesterol, what these lipid, protein, everything in biomolecules, which is the very next chapter. Don't worry about it. But for now, I'm just explaining briefly. Okay, so the ratio of protein and lipid varies considerably in different types of cells, obviously. And in since why is it obvious? That's because all eukaryotic cells are not identical. That was already mentioned. Since they're so complex in nature, different types of cells, different type of eukaryotic cells will have different composition as well. Depending on the type of function that they are performing, they'll have different composition as well. Okay, so the ratio of protein and lipid varies considerably in different types of cells. In human beings, the membrane of erythrocyte, which is RBC, another way of saying RBC is erythrocyte, has approximately 52% protein and 40% lipid. Okay, so depending on the ease of extraction, means how easily can I extract something, depending on the ease of extraction, membrane proteins can be classified as integral or peripheral. Just by the words itself, you can understand what is this classification, like what do they mean by this classification, right? Because if it is integral, it will be totally integrated in the cell membrane, whereas if it is peripheral, it will only be on the top layer of the or the bottom layer of the, it can be easily removed from the cell membrane. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the cell membrane. Okay, this is a good diagram of the cell membrane, and these are proteins. All these orange parts are the proteins, and this is an integral protein because it is totally integrated in the, this one is an integral protein because it is totally integrated in the cell membrane. So if this is the cell membrane, if the protein is completely integrating like this, if the protein is completely integrating, if this is the cell membrane, if it is a bilayer and the protein is like this, I can't easily remove it, right? I can't easily remove. But if the protein is just present like this, then I can simply easily remove it. There's nothing. So in this case, it would be a peripheral protein, whereas in this case, it would be an integral protein. Okay, so now one more thing I need you to notice here is that you can clearly see the bilayer present here with the polar head outwards and the lipid part of the phospholipid towards the inner part of the cell membrane, as I already explained why that is the case. Then you can also see that the carbohydrates or the sugar, see sugar, it's here it is mentioned, it's very confusing because any normal person who is reading this will first read cell the unit of life and then move on to biomolecules. But here they are saying sugar, and we don't even know where did they say sugar, but actually carbohydrates are a form of sugars. That is why they are thinking that we know, but we don't know. But I am here to explain. See, sugars, as mentioned here, is essentially the same thing as carbohydrates. I need to say something, that you'll notice that these sugars or these carbohydrates are present only on one side of the cell membrane and not present on the inner side of the cell membrane. Okay, these sugars are only present on one side of the cell membrane, not present on the inner side of the cell membrane, and that is why plasma membrane is also has another name. It is the asymmetric membrane, is also called as plasma membrane. Why is it asymmetric? Because it has sugars only on one side of the plasma membrane. Moving on. Okay. An improved model of the structure of the cell membrane was proposed by Sir Nicholson and Singer in 1972, widely accepted as the fluid mosaic model. Okay, fluid mosaic model. Now, mosaic is essentially a pattern. Mosaic is just a pattern, and fluid mosaic model we'll understand what it is. According to this, the quasi-fluid nature, quasi means almost, okay. So quasi-fluid nature means almost fluid nature of lipid enables the lateral movement of protein within the overall bilayer. Okay, so this ability to move within the cell membrane is measured as its fluidity. So now they have explained why it is the fluid mosaic model. Why is it called the fluid mosaic model? Fluid because of the quasi-fluid, means almost a fluid nature of the lipid, which allows the lateral movement of the protein within the cell membrane. So this integral protein or these proteins can actually move within the cell membrane, and that movement is measured by the fluidity, and hence plasma membrane is also called a fluid. Fluid means it's called a quasi-fluid state, right? So the model is fluid mosaic model. Fluid because it allows the lateral movement within the cell membrane itself, and mosaic, as I said, is just a pattern. So random pattern, if you've seen tiles, you just go on Google and search the mosaic pattern, you will see there is this random pattern. So you'll see a particular color background and random pattern, random dots on that particular background. So similarly, in plasma membrane, you'll see polar heads, and on that randomly scattered proteins are present, and this pattern looks like a mosaic pattern, and that is why the name of this model of plasma membrane is called the fluid mosaic model of plasma membrane. Okay. The fluid nature of the membrane is also very important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. Okay, so this is obvious. Cell growth, why is the fluid nature important in cell growth? Well, obviously, if the cell wants to grow, and if the the cell membrane is not in a fluid nature and is in a very rigid nature, how can the cell grow? It can't grow, right? Because it is rigid, it can't increase in size. But here it can increase in size because of the fluid nature of the cell membrane. Then it can form intercellular junctions, and it gets secretion, endocytosis, and cell division. Okay, now what is endocytosis? Endocytosis is essentially, just break down the word endocyto. Okay, endo means taking into, and cyto means cytoplasm or the cell. So taking into the cell is endocytosis. If you're taking something from, if the cell is taking anything from the environment into the cell, that process is called endocytosis. And cell division. So why cell division? Again, it is obvious. When it has to divide, it has to pinch off. It has to pinch off the cell membrane also, right? It has to pinch off the cell membrane into its two daughter cells. That is how it will break. Okay. So that is why the fluid nature is very important. We are going to go in depth into cell division in the coming chapter, which is a very beautiful cell cycle and cell division. Very good chapter. One of the most important functions of the plasma membrane is the transport of molecules across it. Okay, that is obvious, right? The membrane is selectively permeable to some molecules present on either side of it. Now, why is that so? I need to explain that. Now see, as I said that the polar molecules only like polar molecules and non-polar molecules only like non-polar molecules. There is a phospholipid bilayer, right? You know there is this phospholipid bilayer. In this bilayer, I'm just drawing this so that you can understand, okay? Okay, so can a non-polar, see this is what are these lipid parts? They're non-polar, meaning no poles are formed because they're hydrocarbon chains, they are non-polar. Okay, can a polar molecule from the environment come and cross this normally? Can it just cross this non-polar part? It can't, right? Because they hate each other, they don't like each other. So a polar molecule can't cross this non-polar bilayer. Okay, so what happens is that it only allows but can what about a non-polar compound? If a non-polar compound tries to come in, then it can easily pass the bilayer, right? It can easily pass the bilayer without any difficulty. Why? Because this is the non-polar part. Non-polar part will allow non-polar part, but it will not allow polar part. Okay, so they're saying that that is why it is selectively permeable. So why is it selectively permeable? Because it only selectively allows molecules, so the cell membrane only selectively allows molecules to pass the cell membrane, and that is why it is called selectively permeable in nature. And many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Okay, without any requirement of energy, it's called passive transport. So neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient. What are neutral solutes? Neutral solutes is just another way of saying non-polar compounds. Neutral solutes are just another way of saying non-polar compounds. Okay, so because why non-polar compound? Because they won't have any charge, right? They're neutral, so they won't have any charge separation within them. So neutral solutes may cross across the gradient. Why? Because the lipid part will allow the non-polar parts to come in, because lipid part themselves are non-polar. So non-polar will allow non-polar. So they will allow neutral and non-polar compounds. So neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient. Now what does along the concentration gradient mean? Meaning from higher concentration to lower concentration, these neutral solutes can pass across the cell membrane. Water may also move across the cell membrane from higher concentration to lower concentration. The movement of water by diffusion is called osmosis. Now there is this one line creates so much confusion, but you have to continue reading. Because why it creates so much confusion? Because I just said that water is a polar compound, and the cell membrane won't allow polar compounds to pass through it, because the bilayer is made up of non-polar compounds, and it will only allow non-polar compounds to pass through it. So how is a polar molecule passing through non-polar bilayer? Well, that is happening because the polar molecule, see, they'll just explain it here. As the polar molecules cannot pass through the non-polar bilayer, they require a carrier protein of membrane to facilitate their transport across the membrane. See, why are these proteins present here? Why are these proteins present here? These proteins are present here to facilitate the transport of polar compounds across the membrane, because see, the cell itself will need polar as well as non-polar molecules as well. It's not that just because the plasma membrane is selectively permeable and does not allow polar molecules across it, the cell does not need polar molecules. No, that's not the case. The cell needs polar as well as non-polar molecules. So to facilitate, non-polar molecules can directly come across the cell membrane across the potential gradient, means from higher to lower. But what about polar molecules? The cell also needs polar molecules. So to facilitate the transport, to provide transport to the polar molecules across the cell membrane, integral proteins, or these proteins are present within the cell membrane. Okay, so that is the function of these proteins. They're telling us the function also. Okay, so osmosis happens across the with the help of a membrane, with the help of a protein, carrier protein. And the specific carrier protein is also called aquaporin. Aquaporin is the specific, it's extra NCRT, extra NCRT information. But the specific carrier protein that allows water to cross the cell membrane is aquaporin. So as polar molecules cannot pass through the non-polar bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, i.e. from lower to higher concentration. So to force, see, water can fall from the hilltop to the hill bottom. So there is from higher point to lower point water can fall, but Can water naturally just jump through a waterfall from lower point to a higher point? That is absurd. That can't happen, right? So for that to happen, however, we need to provide energy to the water, right? If we ourselves are taking a bucket of water from the downstream and pouring it upstream, then that is a different case. We are giving energy to it. So such transport is an energy dependent process in which ATP, which is adenosine triphosphate, which is a currency of energy, in which ATP is utilized and is called active transport. So such a transport which occurs across the against the potential gradients, which is from higher concentration to from lower concentration to higher concentration, which is against the potential gradient. For such transport to occur, you need energy, and that such a transport is called active transport. Okay, so now let's just I'll just explain this whole paragraph in a table, okay, so that it is easy for you to remember. Okay, types of transport across the cell membrane. Okay, so first you can divide it into active and passive. First you can divide it into active and passive. Passive does not require no energy, it does not require any energy, and it also occurs along the potential gradient. Okay, it requires no energy, passive requires no energy and occurs along the potential gradient. Then you can think about active transport. Active transport requires energy and occurs against potential gradient. Okay, then you can further divide the passive transport into two types, which is simple diffusion, and then there is the facilitated diffusion. So what is simple diffusion? Simple diffusion is that this diffusion or the passive transport of non-polar compounds, and it occurs directly across plasma membrane. Okay, but what about facilitated diffusion? It is for polar compounds and it needs carrier proteins for transport across the cell membrane. Okay, example of active transport is sodium and potassium ion pumps. Okay, so that was it for this video. In the next video we are going to go in depth into what cell wall is. I hope you understood whatever I explained in this episode. Thank you.